Hi guys, welcome to the lecture for lymph node and thymus, as well as the spleen and mouth. Is it right? So let's get into the lecture. So we're gonna start with the lymph tissue. So it is a specialized connective tissue that consists of lymphocytes, right, which are supported by connective tissue format that can be, especially that can be reticular fibers. And we remember from connective tissue that reticular fiber is collagen type 3, right? And we also have, since we are talking of connective tissue cells, inside inside the connective tissue, we have connective tissue cells that are macrophages, reticular cells, dendritic cells, reticular cells. Is it? So we have two types of lymphocytes. We have a B lymphocytes mature in the bone marrow, T lymphocytes which mature in the thymus. So the B lymphocytes they mature and become immunocompetent in the bone marrow, while the T lymphocytes mature in the thymus. So therefore, these uh, qualify the bone marrow and the thymus to be primary lymphoid tissues, primary lymphoid organs. You understand? Now, mature lymphocytes of B and T, um, either B and T, right? They they move from primary lymphoid organs and then they go to secondary lymphoid organs. That's your lymph nodes, that's your tonsils, that's your spleen. You understand now? So primary lymphoid organs, what we have with thymus and the bone marrow. What about secondary lymphoid organ? We have a lymph node, tonsils, and spleen. You understand now? Uh-huh. So the lymphocytes they're involved in cellular, for cellular immune response. That's for the T cells. For B cells, they have a humoral immune response. Is it right? So lymphocytes. So T cells and B cells they protect the body against antigens. What is an antigen? An antigen is anything the body recognizes as foreign. So that can be a bacteria, that can be a toxin, that can be a virus, that can be a mismatched red blood cell or cancer cells. Anything that is perceived as foreign by the body, it is referred to as an antigen. So we have this antigen, they will be bound by what you call antigen presenting cells, and then these antigen presenting cells, like the macrophages, they will hand over the antigen to the T cells or the B cells to undergo a reaction. T cells. T cells manage immune response. What they do, they attack and they destroy foreign cells. They have different mechanisms. We have T helper cells and cytotoxic T cells. So they have a different mechanism of destroying and attacking foreign cells. As for the B cells they have that humoral immune response. What they do, they produce what we call, they mature to become plasma cells, and then they secrete antibodies, right? And these antibodies, they will immobilize antigen, they will enter the complement system, opsonize antigen, and so forth. What about the macrophages? So we have the three main antigen um, presenting cells: macrophages, dendritic cells, reticular cells. These are called the antigen presenting cells, the APC, right? So these macrophages, their function, the phagocyte, the under, they, they, they phagocytose foreign substances, right, and present the antigen to T helper cells, right, for recognition. The dendritic cells, they are spiny looking cells with functions similar to macrophages. They also do phagocytosis. While reticular cells, these are fibroblast-like cells that are responsible to produce reticular fibers, which form the stroma and supports other cells in the lymphatic tissue. All right. So let's now take a look at the lymphoid tissue. Lymphoid tissue is categorized into three. We have diffuse lymphatic tissue, lymphatic nodules, and lymphatic organs. Diffuse lymphatic tissue, This one is just lymphatic tissue that is aggregated in the mucosa of the GIT, mucosa of the respiratory system, mucosa of the genitourinary tract, and they are not surrounded by any capsule. These lymphoid tissue, they are just aggregated, right? So these are also called mucosa associated lymphatic tissue, right? So we have a gout in the GIT, right? Gastro associated lymphatic um, tissue. We have bronchial associated lymphatic tissue in the respiratory system, and so forth. You understand now? We go to lymphatic nodules. Lymphatic nodules, right? They are oval shaped masses, right? They can be found in clusters as as though in mouth, or they can be found just uh, in as just singly, right? Just as a single uh, lymphatic nodule, right? So in the GIT, they'll be found these lymphatic nodules. We have the tonsils, pious patches, the appendix lymphatic nodules. You're gonna see as we go. Then another one we have is the lymphatic organs. So when you talk about organs, you're talking about the capsule present. So you have a dense, irregular connective tissue forming the capsule. Right. So now we are talking about those. That's what we are going to focus on: lymphatic organs, the capsule. We are going to focus on the lymph nodules. We are lymph nodes, spleen, thymus. Right. So lymph nodules, like I said, these are aggregations of T cells and B cells supported by reticular fibers, type 3 collagen, right? They may have, so these nodules, they are they are they are they are classified into primary and secondary. Primary, it will be just uh, an aggregation of T lymphocytes, right, with no germinal center. Whenever you find a germinal center, that becomes a secondary lymphoid nodule. You understand now? So whenever there's a germinal center, that's a secondary. Within the germinal center, that's where the site of proliferation and the maturation of a B lymphocytes into plasma cells. So the germinal cells, we are talking about within the germinal cells, we have the maturation of the B cells. In the periphery, we have the T lymphocytes, right? That form that mantle or crown or the most peripheral portion of the nodule, and that will be deeply stained. The germinal center is a slightly or lightly stained, right? So you can actually appreciate from this histological feature. So this deeply stained is your periphery or your corona or your mantle that will be having T cells, T lymphocytes. This whitish or a lessly stained portion is going to be your germinal center, germinal center, germinal center. You can actually see. So within the germinal center, that's where you have the maturation of the B lymphocytes into mature plasma cells. So this is another one you can actually see here. I can actually understand that this is your non-keratinized squamous epithelia. So this can be your mouth, right? This can be tonsils, right? This can be tonsils, right? Was tonsils you actually see this epithelia, squamous, non-keratinized epithelia. So you can actually see that there is no germinal center here. This is just an aggregation of T lymphocytes, right? Which means this is a primary lymphatic nodule, right? So mucosa associated lymphatic tissue, the tonsils. So actually, what makes or what helps you to see that this is a tonsil, you look at the epithelium. The epithelium, because it will be at the walls of the lumen of the oral cavity. So from the understanding that the oral cavity is stratified, non-keratinized squamous epithelium. So whenever you see stratified, squamous, non-keratinized epithelia, and then you see lymphatic tissue, that is a clear sign for a tonsil. So this is stratified, non-keratinized, squamous epithelia, and then you can actually see secondary and primary aggregations, right? If I'm to look here, this is definitely is going to be a secondary lymphoid nodule because it has a germinal center for the proliferation of B cells and a periphery for the T cells. Understood? Right? Another one here, another one here, another one here, another one here, another one here. Yeah. So this is your tonsil. Let's take a look at the thymus gland. Right, gross anatomically, the thymus gland in young, um, before puberty or up to the age of puberty, it will be found in the superior mediastinum and the anterior mediastinum of the mediastinum, right? But it's going to involute and replace by adipose tissue as you increase in age, right? So it is said to be a lymphoepithelial organ because it has dual origin, right? Dual origin, we're going to talk. So it has a lympho portion and an epithelial portion. So you need to understand where is the lymphoid coming from, where is the epithelial coming from, right? So a lymphoepithelial organ located behind the sternum consists of a lymphocyte suspended by the stroma. Take note, in the stroma, there is they cons uh, they consist what you call stellate epithelial reticular cells. We have no reticular fibers in the thymus. NB, no reticular fibers. Typical MCQ question. Right. So you're gonna find that the thymus gland, right, is going to have a capsule, dense, irregular capsule, and then that capsule it divides into lobes, right? And then each lobe is going to be divided into incomplete lobules by the connective tissue trabeculae, as you're going to see. So this capsule is going to send trabeculae, right? Trabeculae, and this trabeculae they are going to divide into incomplete lobules. Incomplete lobules. So what was the development of the thymus? The thymus is dual development, dual origin. Right. So since we said it's a lymphoepithelial organ, the lymphoid part of it, the T cells, they come from the mesoderm, right? Mesoderm, it comes from the mesoderm, right? But the epithelia, it comes from the second and the third, some say the third and the fourth, pharyngeal pouches of the endoderm. Pharyngeal pouches of the endoderm. The second and the third pharyngeal pouches of the endoderm um, forms the epithelia. The lymphoid come from the bone marrow, which is coming from the mesoderm. So the thymus gland originates from the endoderm and the mesoderm. True. At infant, it is concipious, right? At puberty, it reaches its maximum size, right? But as you now continue to grow, it decreases in size, it involutes and replaced by adipose tissue. Function. The function of the thymus gland is to is for the maturation of the T cells, maturation and the differentiation of the T cells, right? So it's very important to take note of that these T cells, as they are coming from the bone marrow, they go to the thymus. In the thymus, they are trained to recognize uh, to recognize foreign bodies, right? Not self foreign bodies or foreign cells or antigen, but not to recognize self. So every T cells that recognize self, it undergoes apoptosis, it dies, because it may bring about autoimmunity or autoimmune diseases. Right. So a thymus it is also an endocrine gland, but it secretes its own hormones that are needed in the maturation of the T cells. So you have the hormones like the thymosin, thymopeptin, right? These will regulate the maturation of the T lymphocytes into immunocompetent cells. So gross anatomically, it looks like this. It is the left and right lobe found in the superior and inferior mediastinum. Blood supply supplied by the inferior thyroid artery, and as well as branches from the internal thoracic artery. So, what makes a thymus, or what helps you to see that this is a thymus? The lobules are incomplete. So you have septa, or what you call interlobular septa, right? That will divide these lobes into sort of like they are incomplete. You can actually see they're incomplete, right? And you have right, you have a darker cortex, which is basophilic, staining due to greater density of lymphoid of lymphoblasts and lymphocytes, right? And then you can actually see right inside this cortex we have what you call the blood thymus barrier. So the blood thymus barrier is going to be made by what you call thymic epithelial cells joined by desmosomes, right, with endothelial cells and the pericytes of capillaries. So remember, we don't want uh, these T lymphocytes to get into the blood, because they are in the process of maturation. They end up attacking self than attacking foreign. So you need a blood thymic barrier. You understand? Right. Then you're also going to have a medulla. So the medulla is going to be central, lightly stained, right? Because it contains fewer but larger mature lymphocytes, right? And inside this medulla, you have something that is only unique to the thymus. That is what you call Hassall corpuscles. I can actually say that this is a Hassall corpuscle. This is another Hassall corpuscle. So these are unique to the thymic medulla. And take note of this: the Hassall corpuscle are made by um, T lymphocytes that are undergoing apoptosis, that are dying. They form this Hassall corpuscle, and these Hassall corpuscle, they actually increase in age. So if you see the medulla that is going to have a lot of Hassall corpuscle, you can actually predict the age. That because they increase in what with age, right? So this is another good diagram to actually illustrate the thymus. So you can actually see incomplete lobule over here, another incomplete lobule over here, another incomplete lobule, but they will be sharing a common medulla. You actually see. So this becomes your cortex, deeply stained cortex here, medulla, 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 right here, lightly stained because of fewer mature lymphocytes, deeply stained because of small lymphoblasts and lymphocytes. You can actually see the trabeculae that we're talking about, dividing uh, the thymus into lobules, incomplete lobules. And you can actually appreciate a capsule right here, a dense irregular connective tissue capsule right here. Right. So let's just take a look at these lobules. So each lobule has a darker cortex and a lighter medulla. Darker cortex, lighter medulla. Darker, darker cortex, lighter medulla. You understand? So the cortex contains numerous T lymphocytes, which are precursor cells, thymocytes, right? We also have macrophages and epithelial reticular cells. Then we have no reticular fibers, we have epithelial reticular cells. These reticular cells, they are joined by tight junctions and desmosomes, right, which also take part in the formation of the blood, uh, uh, the, the thymic blood barrier, right? So the thymocytes, they are actually just trained to mark foreign from self, right? So they mature into two types of T cells. We have T helper cells and cytotoxic T cells. And their maturation is going to be mediated by those hormones, the thymosin and the thymopeptin, right? And these hormones, they are produced by those epithelial cells, which were coming from the second and the third pharyngeal pouch of the endoderm. Take note of this. The medulla of the thymus contains epithelial reticular fibers, mature T lymphocytes, that's why it's lightly stained, and thymic Hassall corpuscles. Take note of this. Hassall corpuscles, they contain flantain epithelial cells filled with keratin filaments. So that's why they are lightly stained. They are eosinophilic because they have what? They have a protein keratin. Right, so you can actually see this is a septa right here. This is your cortex right here. Actually see it has a corpuscle right here, some microphages right here. Um, actually see another microphage right here, microphage right here. You can actually see this star-like shape. These are your epithelial reticular cells or your thymic epithelial cells, right? The ones that will form the stroma, right? Forming a compartment that is called a cytoreticulum. So this is a cytoreticulum, cytoreticulum. These are just compartments formed by these cells. Another good diagram here. So you can actually see this is your periphery, your outer darker cortex, your inner medulla. You can actually see the has a corpuscle, has a corpuscle, has a corpuscle. They are very eosinophilic. Why? Because of the presence of keratin. Your trabeculae, and in the trabeculae, you're also going to have blood capillaries, actually blood capillaries in this trabeculae, right? So you can actually now see that the lymphocytes they have a large nucleus, and these pale cells, right? The small nucleus, they're definitely going to be your reticular cells, right? You can actually now see this is a concentric folds of keratin and dead uh, T lymphocytes forming this aggregated uh, corpuscle called the Hassall corpuscle that we said it is found and unique in the medulla, and as well as um, increase its number as you grow, right? So I can actually say this is a T lymphocyte, this is another T lymphocyte, this is another T lymphocyte, this is a, what is this? This is a reticular cell, right? Reticular cell, another reticular cell, T lymphocytes. Yeah. Now let's end by talking about the thymic blood barrier. So the thymic blood barrier, it consists of continuous endothelium of the thymic. Whenever there's a barrier, you have to think of continuous basal lamina, right? With the epithelium, with the endothelium joined by tight junctions. And also